Hello and welcome, dear friends. Today let's take a look into the world of gene. What does it mean when we say something as operon, and what does it mean when we talk about gene regulation? So this lecture is a primer for understanding the complete unit of regulation of gene expression. Gene expression basically means protein synthesis. Means gene expression can be synonymously used with the term protein synthesis. So when you say a gene is getting expressed, we talk about a protein being synthesized. A protein which may have a catalytic function and may act as an enzyme, and which may mediate all the metabolic pathways in a cell. So today we will see basically what is a gene, what is a operon, and what is gene regulation. So before going into the concept of the function of a gene or the terminologies associated with a gene, let us just understand what is a chromosome and how is it different from the term DNA and the terminology gene. Genetics is a branch of science which deals with the study of genes, which deals with the study of DNA. And DNA, you know, is the basic hereditary material of all the living cells. Now we have across this living kingdom two broadly categorized systems, the eukaryotic system and the prokaryotic system. So on the screen on my left there is chromosome concept, and on the right there is a bacterial chromosome concept or the bacterial DNA concept. When we speak about a bacteria, we don't call it as a bacterial chromosome as it is written here on the screen, but it is not wrong to say that. We can call it as a bacterial chromosome. And how or what is the difference between a chromosome and a DNA, we are going to see in this slide. Now let us start with the DNA. Both the pictures you can see on my right, this is bacteria and this is a eukaryotic chromosome, which is a much more complex and organized system as compared to the primitive nature of bacterial DNA. So if you see the DNA, which is around 2 nanometer across here, now this double stranded DNA, follow the red LED cursor here. So this double stranded DNA is coiled. First of all, as you can see, this is a double stranded linear DNA molecule in eukaryotes, in the multicellular system, in plants and animals. You can see it is coiled around special proteins called as histones. There is no such thing in the bacteria. I will come to the bacteria afterwards, but just see the beauty and the complexity of the eukaryotic chromosome organization. Okay, so you can see this is the first binding around histones. So this is structure of DNA bounded around the histones is called as chromatin, and this is found by the winding of the DNA around histone proteins. One thing, right? Then there is a sort of structural hierarchy as it goes on becoming more and more complex. You can see very systematically the wound DNA around the proteins accumulates to form a chromatin structure, okay, which is again a double stranded DNA wound around histines compacted, and that histine protein becomes a thread like structure, and a secondary structure comprising of the DNA loop linked by special proteins called as you can see, two proteins I have shown here. This is condensin protein and this is cohesin protein. All this is in eukaryotes. So this strand of DNA wound around histones, which looks like again a very single thread, now that is interlinked with another, it's systematic looping, which will be later on we will call it as chromatids, and the single strand is called as chromatin. Single, it is not single, it is double stranded DNA wound around the histones, very systematically compactly arranged, and that arrangement means the chromatin, that is DNA around histones, is then again loop linked by condensin protein. Now this is condensin, you can see here, I will try to zoom it, see here. Now this is the zoomed structure. You see this is a chromatin structure wound around condensin. Now imagine this condensin as the hairpins, okay, hairpins which are used by the ladies. Now this is just like one big pin and this cohesin is a small pin. Now condensin, many molecules of condensin, you can see here, this is one structure and this is 1, 2, 3, 4, 5, 6, 7, 8 condensin structures rounded off, and 8 loops are there around which systematically the chromatin fiber is interwound around each other. So two such condensins are then in the middle, you can see, are interlinked by cohesin. Okay, so the double stranded DNA wound around histones, then histone structure will be called as chromatin. Chromatin will be looped around, that is loop linked into condensin proteins, a beautiful structure. Two condensins are then interlinked with cohesin, again a protein molecule, and like this you go on to form several units of such condensins. They form chromatid. This is a chromatid, that is one arm, you can say, of the chromosome. So sister chromatid, that will be called as, is a structure which is made up of many densely condensed condensin units, and then this condenses into a chromosome, and these chromosomes inside, that is, it forms a structure with a centromere, and sister chromatids, they produce the DNA, replicated DNA structures, and this is what you have, a eukaryotic chromosome. So if you come reverse, a centromere, that is the chromosome, if you open it you will get sister chromatids. Sister chromatids are structures where the structure is made up of densely condensed, systematically looped condensin structures, and you open it up, you will get chromatin structures, that is DNA wound around histone proteins. You open up, remove the histones, and you will get a double stranded DNA. So this double stranded DNA molecule which is there, this DNA, if you come and see it in the bacteria, it is a free covalently closed circular molecule. Let us zoom into this again. Bacterial structure. See, the bacterial structure is simple, it is primitive, and what actually happens with this structure is, it is freely present in the cytoplasm. Extra chromosomal material like plasmids are associated along with it in many bacteria. Then these bacterial chromosomes, these, if you open up directly, no histones, no chromatin, no cohesin or condensin, nothing of such. DNA is not associated with any of the protein molecules. It is freely present in the cytoplasm, and you open up it up into a double stranded DNA molecule, simple structure. And we are trying to study the gene expression with this simple structure, because whatever the gene expression patterns, these are present in the bacteria, similar type of patterns or the mechanisms exist in the eukaryotic system. Only the difference is that it is a nuclear membrane bound DNA, so the transcription is taking place within the nucleus and translation takes place outside the nucleus in the cytoplasm in eukaryotes, whereas in prokaryotes it's a coupled transcription and translation process, as we saw in the earlier lectures. So this is the complexity of DNA. So what do we know when we say a chromosome? A chromosome usually is a compact structure. Here also, this compact, covalently coiled, closed, of course not associated with any proteins in the bacteria, that compact covalently closed structure can be called as bacterial chromosome. As you go on opening it into a double stranded, simple single double stranded molecule, that segment will be called as DNA, or that double stranded region will be called as DNA, and in that a particular region which is responsible for synthesis of a protein will be called as a gene. So gene is a segment of a DNA. DNA is a linear part of the chromosome, a free part of the chromosome which is free of any associated proteins. So in the bacteria you can call it as bacterial DNA directly. Rather than calling it as a bacterial chromosome, you can call it as a bacterial DNA, because it is not associated with any proteins. Whereas in eukaryotes, a very systematic, beautiful, symmetrical and nicely arranged DNA, because it is huge in size. In eukaryotes we have sets of chromosome, diploidy is there. whereas this is haploid, single, freely available in the cytoplasm, so on and so forth. So this is the complexity, comparison and contrasting of a prokaryotic DNA as well as with a eukaryotic DNA and a chromosome, concept of a chromosome, DNA and a gene. So the organization of eukaryotic chromosomes is much more complex. So if you see just from the bottom structure here, this is a chromosome, centromeres are there, just one single region, see the rectangle here. If you just magnify this simple part, it further condensed chromatin you can see, that is which contains cohesin, which contains the molecules which are interlinking the chromatids, etc. That single part of condensed chromatid, if you take this rectangle, magnify, you will find nucleosomes, a coiled chromatin fiber, that is double stranded DNA wound around histone proteins, and histones systematically organized to form chromatins. Then again a small part of this chromatin, if you magnify, you will find that this DNA is wrapped around a special protein called as histone, and that simple single part of that DNA wrapped around histone, if you see, you get our Watson and Crick's double stranded DNA molecule. This is how the eukaryotic chromosome is organized. So double stranded DNA is wrapped around histones. Histones, they are systematically supercoiled again, and they are again interlinked by various special types of cohesin and attachment proteins. So they form a chromatin fiber structure. They are further condensed and it forms a chromosome. So this is the complexity of a eukaryotic chromosome. Strands of DNA, they wrap around histone forming nucleosomes. Nucleosomes, then they coils together to form chromatin. Chromatin loops and coils to form supercoils. Supercoil bunch together to form chromosomes. So this supercoiling is carried out by systematic organization by cohesin and condensin proteins. This is after the coiling here, this supercoil is by condensin and cohesin proteins. But if you see in the prokaryotic cell, this prokaryotic DNA, it is a chromosome itself, which has one single point of attachment to the nuclear membrane. If you see here, I will zoom it, you can see the point of attachment here. This is the point of attachment of one single point. It is attached to the nuclear membrane or the cell wall, which gives some sort of stability of this DNA. Otherwise the rest of the whole circular DNA is covalently closed, supercoiled and freely floating in the cytoplasm in the prokaryotic system, whereas eukaryote, we have just seen, it is very systematically organized. So this is how the prokaryotic and the eukaryotic DNA, they are organized. So the concept of a chromosome as well as a gene, a chromosome is the most condensed structure of a DNA molecule which is associated along with proteins. Remember, how will you categorize or how will you define a chromosome? A chromosome is the most condensed structure of a DNA molecule associated with proteins, whereas gene is a locus. Locus means a point on the chromosome. The chromosome is always made up of DNA, whereas a gene is can be made up of a DNA or a RNA. We can even call a segment of RNA which is responsible for synthesis of a protein as a gene. On the chromosome, mutations are relatively very large because there is a diploidy, and on the homologous chromosome there are mutations, whereas mutations are somewhat smaller in size as compared to the chromosomal in the gene structure, and because it occurs either only in replication or when there is a DNA damage, as we have seen in our past lectures. So this is how the eukaryotic and the prokaryotic cell harbor the DNA, and that is the magnitude or the complexity of the genome. Now, having said that, I would like to emphasize here that eukaryotic cell genome and the prokaryotic cell genome show almost the equal magnitude of metabolic activities, means they are no more no less when compared to their function in terms of metabolism. Now the function of a gene. What is a gene? Gene is defined, as I said, a locus on the chromosome. We will see some structure and function of genes also. Modern concepts of gene we will see in this lecture. So basically, when you talk about a gene, what is the function of a gene? The function of a gene is protein synthesis. So this is a eukaryotic chromosome. If you want to talk about a bacterial chromosome, start from here, because there is no association. This DNA, this can be called as a bacterial one. If you want to talk about a eukaryotic chromosome, then this is a compact, cohesin, condensin, chromatin, chromatid, organized chromosome of a eukaryote. Anyway, whatever, that DNA in both is same, double stranded DNA, and that part of the DNA which is responsible for synthesis of one particular protein, one particular enzyme or a hormone or whatever the metabolic activity it is responsible for, genetic activity or a metabolic activity, that segment will be called as a gene. And that gene is therefore in very simple term defined as a functional unit of the DNA. Gene is called as a functional unit of a DNA. What is the function? The function is protein synthesis. So the function is DNA, that gene, that length of the DNA which has a initiation point and a termination point, that in between that region which is responsible for some function, that function is formation of a protein, is called as a gene. So gene is a functional unit of a chromosome. The function is transcription, translation and formation of a protein. This is what the central dogma of gene expression, central dogma of protein synthesis, central dogma of genetic functions, central dogma of life is all about. So we can use it alternatively. So I will call it here central dogma of gene expression. So the function of a gene, as you can see, I have shown this figure. This chromosome, it is made up of 10⁻⁷ to 10⁻⁻10 base pairs, whereas a gene is around 10⁻³ to 10⁻⁶ base pairs. And the function is, it will synthesize mRNA by transcription, which will be translated into protein. That protein may function as an enzyme, and that enzyme is responsible for a variety of metabolic activities. Apart from that, the gene may also produce a non-productive, that is non-protein product, which is RNA, and that RNA can function for protein synthesis. Like there are three types of RNAs: mRNA, rRNA and tRNA. So these mRNA, rRNA and tRNA are also the function of a gene, but they are somewhat stable structures, not constantly synthesized. And it is not that a gene is always responsible for synthesis of a protein, not always. There are three functions of a gene. One, there is a gene which does not synthesize anything, but it allows the attachment of a protein. I will explain what type of gene that is. Second is a gene which results into a protein synthesis, that is called as a structural gene. And there are genes which are responsible for not for the formation of proteins, but for the formation of stable structures like ribosomes, tRNA and mRNA. Okay, so that is how the central dogma of protein of gene expression functions. DNA, if it replicates, it forms a new DNA. DNA, if it transcribes, it forms a RNA, and if it translates, it forms the workhouse of the workforce chemicals of a cell, that is proteins, enzymes, vitamins, cofactors, etc. So that is how it is the function of a gene is all about. So the point here is that this figure should summarize, or if you see this is an artistic representation of the concept. The bacterial and the eukaryotic genome, they are the same when it comes to the universality of the code. We have seen in our previous lectures the characteristic features of a code, and universally speaking, genetic code is universal in nature, means it codes for the same types of amino acids, it codes for the same types of that is 64 codons are there. Code is degenerate, the code is not having any comma. There are stop codons, there are nonsense codons. All these are same even if you are considering about eukaryotic DNA or a prokaryotic DNA. And what is the function? The function is protein synthesis, that is ribosomes and protein synthesis. So the function, therefore genetic engineering is possible. You can use a prokaryotic DNA to encode in a eukaryotic cell and vice versa. That is made possible only because of the universality of the code, and that is the function of a gene. So function of a gene is protein synthesis, and protein synthesis is the way by which a gene expresses itself. So that is gene expression. Now let us see some theoretical aspects. for you to take down the notes or understand the jargon in which it has to be mentioned. Gene, first of all, the gene is defined as a segment of a DNA which is responsible for inheriting phenotypic and genotypic character, which is in general called as inheritance, and also responsible for expression of a character, that is a physical or a genotypic trait. A gene is a segment of a DNA which provides instructions for the synthesis of a specific protein or a particular type of RNA. Gregor Mendel was the first to call genes as a unit of inheritance, and he called them as factors. He used the term factors for gene because they are responsible for synthesis of proteins. The term gene was derived from the Greek word genesis. Genesis means to be born, and this was coined by Danish genetist William Johnson in 1909. Some of the characteristic features and the functions of the genes we will see. Characteristic features of the genes are functional unit of heredity. They are responsible for variation, mutation and evolution of all the living systems on this earth. Genes, they determine the physical as well as physiological characteristics of the organism. Genes are responsible for transferring of these characters from parents to their progeny, that is the offsprings, from one generation to the another generation, and that is what genetic study is all about. Genes are situated in the chromosomes. Every gene occupies a fixed position on the chromosome, and this position is called as loci or locus. The most important function of gene is to express themselves by synthesizing proteins. Proteins which may be acting as enzymes, and they control the cell metabolism. Thus they determine the physical and metabolic characteristics of the cell. Okay, each and every metabolic and physical nature of the cell is by virtue of genes. Each gene, it synthesizes a protein which acts as an enzyme, and then it brings about the appropriate physico-chemical and the biological change in that system. These genes, they can be duplicated, replicated automatically when the DNA replication takes place, and a single gene may have its variable when it is in the eukaryotic system, or even temporary diploids may be there in prokaryotes also, and when the similar type of gene is available on another extra chromosomal entity like plasmids, that will be called as a allele. Means on the chromosome there is a gene. For example, lactose utilizing gene is present on the Escherichia coli chromosome, and the same gene can be present on a plasmid also. So both these genes in two copies, they will be called as alleles. Okay, so these genes, they may have their alternative forms which are called as alleles, and sometimes what happens, there is one lactose gene. These both can be dominant, both can be recessive, one can be dominant, one can be recessive, one can be dominant, this can be recessive. So recessive and dominant factors predominate the alleles. So this is the characteristic features of a gene. Escherichia coli, now if you see the complexity, means the magnitude of genes, E. coli has about 4,400 genes, whereas humans, they have around 30 to 40,000 genes. Okay, so you can see huge number of 10 times we have more genes as compared to the bacteria. But the problem is we have a lot of junk DNA which never functions. Maybe that have been silenced during the evolution, or maybe they will function somewhere near in the future. No one knows that. So the modern concept of a gene, what is the modern concept? Now today we don't just call each and every gene, we have given it a specific function. Like if it is a gene responsible for recombination, we call it as a recon. If there is a mutation undergoing in a particular gene, and that gene, like transposons, they are continuously mutating, we call it as a muton. So Benzer in 1955, he introduced the term cistron, muton and recon. Cistron means just like the cis and trans effects of a molecule, this gene has two dual nature. One is it is responsible for synthesis of a protein, which is called as the cis version or the trans version, that is a gene which does not form the protein, but it is allowing the attachment of a protein. So we will see that in the next slides. So the modern concept of a gene can be called as cistron, which is the unit of a function, muton, which is unit of mutation. Means cistron is a segment of DNA which is having a particular function, that is for expression of a protein or a particular trait. Muton is a segment which of DNA which undergoes mutations. It can be few base pairs long. Recon is that gene which is responsible for transcription, translation, transduction or transposition type of recombination. Operon, this is our main area of focus. Operon is the combination of or the structure of a gene which has operator, promoter, structural genes, regulatory genes, which I am going to explain in the further slides. And replicon is that gene which is responsible for replication. The functions of a gene, you know very well. These genes, they are responsible for proteins, and you can see here in the figure a protein A, B, C. These are transcribed from three genes, gene A, B and C. These three A, B, C genes may be present attached in tandem to one another or may be present at different loci on the chromosome, but these three proteins can combine to form a tertiary type of protein, a more complex type of protein. I will give you the example of this type of organization of proteins from different gene coming close together to form a three-dimensional protein which is functional for the cell. So what are the functions of the genes? Functions of the gene is to maintain variation, heredity, mutation, etc. Genes, they control the phenotype of the offspring, means both the structural and the functional characters. Genes control the reproduction and the replication of the DNA also. Genes, they are the ones which undergo mutation to give rise to polymorphism or mutations which become the raw material for evolution. That all things takes place in the genes. Genes are associated in the eukaryotic process, eukaryotic systems with aging process. Genes are responsible for producing cancer also. The genes, they are controlled, regulated, and the transcription of mRNA and the translation of protein synthesis is controlled, means how much amount of a protein has to be synthesized is that decision is made on the regulatory sites on the gene. That will be the discussion in my next lecture, regulation of a gene. Regulation of a gene means how does a gene decide the quantity of a protein to be synthesized, means when does it decide to turn the gene system on, gene expression on and off. The capacity of a gene to regulate the on and off of protein synthesis is called as gene regulation. So the genes also, they code for different types of RNAs, that is mRNA, rRNA, tRNA. Genes are responsible for regulation. This is one of the important aspects of the discussion which we are trying to understand in this unit. So genes, they control the function of other genes also, and therefore genes are responsible for the overall development of a cell. So this is in general a function of a typical gene which we will call it as a operon. Operon is a operational gene. So you can see there is a promoter on the DNA which is responsible for binding up of a polymerase, RNA polymerase. So this becomes a regulatory gene, means this gene, promoter gene, is not responsible for synthesis of a protein but is responsible for attachment of a protein. So it is called as the regulatory region. So as the regulatory gene attaches the RNA polymerase, it goes on synthesizing mRNA. This mRNA will synthesize a polypeptide or a protein. So that region of the gene which is responsible for this polypeptide is called as structural gene, and there will be lastly there will be gene sequences which are responsible for termination of the protein synthesis. So this is in general the operon. Operon, for a more detailed concept of operon, let us see what is the operon looks like. But before that, as I said, here you see in this example, genes are connected to other genes, and gene A, B, C products, protein A, B, C, they come together to form a complex protein. Now one best example which you have all studied is immunoglobulins. Now you see there is no need to worry much about this slide or the complexity of this slide. You know very well immunoglobulin molecule is having light chain. Here this is the light chain and the heavy chain. The arm, small arm is the light chain and the Y-shaped molecule is the heavy chain. Now all these heavy chain and light chain molecules, these are synthesized from the somatic germline DNA. So there is DNA, then that is transcription, there is translation, and there is a polypeptide chain of the germline DNA. This polypeptide chain are synthesized from 1, 2, 3, 4, 5, 6 regions, and then it forms, and splitting and organizing forms a light chain. Then from some other region of the DNA, you can see here there is a heavy chain synthesis which is spliced, joined to the light chain, and you have immunoglobulin molecule. So immunoglobulin is a complex protein which is a product of protein synthesis or gene expression from various regions of the DNA to form one immunoglobulin molecule. Like that, there are many classes of immunoglobulin molecules you can see. The same thing happens in the bacteria when it comes to flagella, which is a protein, or the enzymes which are proteins, so on and so forth. So what is this Operon concept? For this Operon concept, you see the gene structure. A prokaryotic gene structure and a eukaryotic gene structure looks like this. In a prokaryotic gene, you have a promoter region where the binding of the RNA polymerase takes place. Then there is coding sequence which is called as the structural gene, and a terminator. Whereas the eukaryotic gene, it is much more complex. It has non-coding sequences, and these are called as introns. Whereas you see in the prokaryotic gene, it is a complete linear DNA molecule which each and every part of this gene is responsible for coding of some or the other molecule of a protein. But in eukaryotic gene structure, there are introns. These introns are encoded on mRNA, means the length of the gene on the DNA results into a length of a gene on mRNA. On the mRNA, these introns are spliced off, the exons are joined, means the final transcript of mRNA is smaller than the DNA gene. And then there is a 5-methylguanosine cap added, and there is a polyadenyl tail added in the eukaryotic protein synthesis during the mRNA, and then this mRNA is translated into protein. So prokaryotic gene, you know, there is a coupled transcription and translation. Eukaryotic gene, there is a separate transcription and a separate translation. So this is the gene structure. Now let us see the operon and its function. The function of a operon is not only to produce the proteins or the enzymes needed, but also to regulate its concentration. Means at what particular given point of time how much protein is required, and when that need for the protein is over, to stop the process. That is the function of operon. Basically means a regulation. Operon will not talk about only the genes which are responsible for protein synthesis. Like this, if you see here in the figure here, now this A, B, C, D are the actual genes. You can see the mRNA, the length of this pink colored mRNA corresponds to A, B, C, D gene. So we see this A, B, C, D, it transcribes into a mRNA, and mRNA, it is responsible, it is a polycistronic mRNA molecule, means a gene which is responsible for synthesis of more than one type of proteins. So this polycistronic mRNA molecule produces protein A, B, C, D. Let us say 4 proteins. One gene responsible for 4 proteins. Now that is all from a single mRNA, because these 4 genes are present one after the another. Means there is a start codon, stop codon, start codon, stop codon, start codon, and stop codon, so on and so forth. So this is called as one single mRNA which encodes for 4 different types of protein. Now this is gene and a protein gene expression concept. But operon concept has other genes associated with it. That is, these are those genes which are not actually forming a part of this product, that is the protein, but they help in regulating the production. Means this regulatory gene, there is a promoter gene, operator gene, and then the structural genes are there. So regulatory genes are those genes which will synthesize some product like a repressor, or it will attach itself to the DNA some protein like a co-repressor or a repressor or activator or an enhancer. We will see that in detail. But a gene consists of some other parts or other segments of the DNA also, which regulates the production of proteins of this particular gene. So that in toto is called as operon. So in prokaryotes, the structural genes of related functions, they are often organized together in the genome, and they are transcribed together under the control of a single promoter. Okay, promoter. So operator, promoter, these are the regions where RNA polymerase and some other proteins which will either block the movement of RNA polymerase or will allow the movement of RNA polymerase. This is these two regions. Okay, so they are regulated, controlled at this point. The operon's regulatory region, this regulatory region, it includes both operator and promoter, and this is the region for the binding of repressor. Repressor binds at the operator and RNA polymerase binds at the promoter. The operator binding repressor may be synthesized from a regulatory gene. So we have very two good beautiful examples of this operon and its function in the form of lac operon and tryptophan operon, which will be coming in very much detail in the next lectures. But in general, whether that is utilization of lactose or production of tryptophan or any other function, each and every gene has a structure like this. Means it has the actual structural genes which are responsible for the proteins, and it has other regulatory genes, operator and a promoter gene also including, which regulates the production. Okay, alternatively there may be some activators also, some proteins which will enhance, they will bind to the regulatory region and they will enhance the transcription, allow the polymerase to move further. So this in general is the structure of operon. The basic concept of operon is a group of functionally related genes coming together, coordinating, and they are controlled, as you saw in the previous figure, 4 proteins controlled by one switch. Come here. So this is, if you call this operator, promoter and regulatory genes as switches, just like you put the light wherever in the building, first floor, second floor, third floor, you can have one switch controlling these lights. So just like this, these are called as genetic switches. These genetic switch, a group of functionally related genes, can be coordinated or controlled by a single on or off switch, and that is called as the operator or the promoter switch. The regulatory switch is usually a segment of a DNA which is called as an operator, and it is near the promoter. It is positioned just near, in the vicinity of the promoter. And the operon is an entire stretch of the DNA which includes the operator, the promoter and the genes which are responsible for protein synthesis. Now if you see here, we have this operator and promoter, two genes which are not actually forming these proteins. You see, the protein synthesis starts from this region of the mRNA, that is region of the A gene. So now we will have these two types of genes in operon. Promoter and operator, they are not responsible for structural proteins, but they help in formation of this protein. They help in binding of the proteins. On promoter, RNA polymerase binds, on operator, a repressor binds. Okay, so these are the genes which are not actually synthesizing proteins but binding certain proteins. So that is one type of a gene, and immediately there are genes which are responsible for protein production. So this is called as a cis operating gene and a trans operating gene. So trans operating gene is that gene which forms, this is the trans genes, A, B, C, D are the trans genes which form a protein, and cis genes are the genes which are responsible for not a functional protein but attachment of a protein. So we have this combined concept called as a cistron, and from the cistron it is the concept operon is developed. So trans acting genes, trans and cis, trans acting genes, a product that can function on any copy of its target DNA. It means that it can form a diffusible protein or a RNA, means there is some product which is formed. And trans acting genes, these are the cis acting genes or cis genes. Trans and cis. Cis genes are, it is not a gene which is responsible for a protein, but it is a site on the DNA which is responsible for, does not code for a protein but codes is there for attachment of a protein like RNA polymerase or repressors. So when come back to our structure, so this is now this mRNA which is formed, it is a product of a cistron. This is the structural genes. This is a operator, promoter and structural genes. This short mRNA is a cistron molecule. Now as this mRNA encodes for more than one type of proteins, that is protein A, B, C, D, one or more than one. If it is only one protein, it will be called as a monocistron, and if there are more than one types of protein, it will be called as a polycistronic mRNA molecule. So that is how the concept of operon is derived. Now operon and its function. If you see, operons may function for regulating the catabolic pathways as well as anabolic pathways. What is operon? Operon is a gene. What is the function of a gene? To produce proteins. What is the function of a protein? To act as enzymes. Where are enzymes required? Enzymes are required for metabolism. Metabolism is made up of two types of metabolic activities, catabolism and anabolism. So there are catabolic pathways where breakdown of substrates takes place. There are anabolic pathways where buildup of system takes place. All these physiological, biological, genetic, phenotypic changes via metabolism is by virtue of either catabolism or anabolism. All these catabolic and anabolic activities require enzymes. Enzymes are synthesized from genes. Genes are functioning as operons. So there will be operons which function for catabolic pathways, and there will be operons which will be responsible for anabolic pathways. Catabolic pathways means breakdown of substrate. We have a very good example of catabolic operon, lactose operon. Utilization of lactose, breaking it down for energy. And there is one beautiful example in your syllabus for anabolic operon, that is the tryptophan synthesis. Amino acid synthesis. When amino acid is not there in the medium, the cell needs it for energy, needs it for formation of proteins. So that is an anabolic pathway. We will see that in our future lectures. So the prokaryotic gene expression, as you know very well, it is a coupled transcription and translation. And I have just shown you this figure where we defined operon, and we will see some more molecular level organization of a gene and the operon. When a operon operates, it operates in two different functions, positive and negative, and either in an inducible or a repressible system. So and a permutation combination of these four. So I will try to explain this as a final part of today's lecture, that is, what is a gene expression all about? How does a protein gets synthesized and is regulated? The core concept. Detailed aspect will be there in the next lecture. So we have seen that the prokaryotic protein synthesis takes place in a coupled fashion. Coupled transcription and translation is there. Whereas the operon is defined as a unit of bacterial gene expression where it regulates the whole process, which includes the structural genes and the control elements like the regulatory genes. Now, regulatory gene in this figure, as you can see, is a gene which codes for one protein. That can be an activator, that can be a repressor. Regulatory genes, these codes for a protein. Promoter and operator do not code for a protein. And the structural genes, they code for the actual enzymes or the protein responsible for metabolism. Whereas regulatory genes codes for those proteins which do not actually take part in the metabolic process, but they help in the formation of proteins which will be responsible for metabolic processes. Okay, means here A, B, C, D proteins which will be formed, that will be controlled by these regulatory proteins. Two proteins are formed, either a repressor or activator. I will show you. It's a very beautiful and interesting concept. I want you to focus on this. So regulatory gene codes for a product which is a protein which controls the expression of other genes. Means there will be some proteins formed here by these regulatory genes which will control the quantity, time and production of ABCD genes. Okay, but they have no role in the structure of ABCD. Then structural genes, as you know very well, this is the gene that codes for the regular enzymes. So what are the products of regulatory gene? This regulatory gene, it codes, of course, there is mRNA, and mRNA is translated into a regulatory protein. This regulatory protein will act at the operator site. On the promoter there will be RNA polymerase, always remember. on the promoter on the DNA, there always will be RNA polymerase which will express these structural genes. Now the question is whether this RNA polymerase on the promoter is allowed to enter into structural genes or not. Now on the operator, if there is a speed breaker, means there is some another protein attached, like you can see there is in the figure here. Let me zoom it. So see here, this is a regulatory gene. In the regulatory gene, it forms a regulatory gene is away from the cluster. It is not associated with the structural gene, or it is away from the operon, you can say. So here it can be near the operon, it can be away from the operon. What is the function of a regulatory gene? To synthesize a protein. Now this protein can be acting in a positive way or a negative way. What is that? I will show you in the next slide. So in a certain way, it binds to the target site. Where does it bind? It typically binds to the operator. No question of promoter. Promoter is always a permanent site for the RNA polymerase. Now RNA polymerase with a promoter is just like a marble on a sliding table. If you place the marble on a sliding table, it will roll down. So if I put a breaker there, then that marble will stop here. Just like that, promoter, if it is containing a RNA polymerase, it will always run into the structural genes. But it has to be halted when it is not needed, and that halt is the region called as operator. And that is halted by a protein, let us call it as a repressor. Repressor is something which represses the promoter, and it is bound at the site of the, that is a target site operator. This is how the regulation takes place. Now gene regulation, which means the terminology gene regulation means the level of genetic expression which varies under different conditions, means which can be controlled at the level of concentration. There are various levels of gene expression. If these gene regulatory genes are not there, then there will be continuous synthesis of ABCD proteins. If the RNA polymerase is not stopped from entering into structural genes, then continuously ABCD, this will be synthesized without really taking into consideration if they are needed or not needed. The question of discussion here is, how does the cell decide when to start a gene expression, when to stop a gene expression? This basic process is called as regulation of a gene expression. Okay, so there are various levels of control. We will see those regulation of control at various levels, but at the core level we have to understand that it can be positively regulated or it can be negatively regulated. Regulation of gene expression, it is very important to note that can be, see the structure here, can be at the level of transcription, can be at the level of translation, can be at the level after translation, that is the post-translational changes. Gene regulation is very important for the cell process to conserve energy, for metabolism, for the environmental response, and the cell growth in general. Regulation can occur at any one point of these gene expression sequences, that is either at the level of transcription it can be there, either at the level of translation gene regulation can occur, or even at the post translation level this can occur. So we will see the basic types of regulation in any type of cell, eukaryotic, prokaryotic. When you think about gene expression, this gene expression is regulated either negatively or positively, or it can be an inducible system or a repressible system. Remember the four terminologies: negative regulation, positive regulation, inducible system, repressible system. What are these four terminologies? And there can be a permutation combination of all these four possibilities, which is the final part of discussion of today's lecture. So you can see here, cis acting means one which is acting on the operator and the promoter. There is operator and a promoter before the structural genes. We have seen the gene organization. Operator and promoter genes are always before the structural genes, because promoter is the site of binding up of a RNA polymerase. So gene, it is attached to the RNA polymerase, it initiates the protein synthesis. Now you see at the operator region there is no blockage. There is no blockage, and this RNA polymerase runs into the structural genes. Once it runs into the structural genes uninhibited, without any stoppage, it forms RNA and the protein. Now the point here is, for how long this will go on? If this protein is not needed for the cell, remember, a cell will never undergo a wasteful synthesis. A cell will never undergo a wasteful synthesis, and it will stop when it is not needed. How it is stopped? It is stopped by the regulatory gene synthesizing a protein called as a repressor, and that repressor binds at the site of the operator. Always there is a repressor bound to the operator, and always there will be RNA polymerase bound to the promoter trying to go into the structural genes, but as it is blocked, it will not enter. So gene is turned off when repressor is bound to the operator. So as it is a negative process, not allowing the RNA polymerase to move into the structural gene, repressing the activity with the help of a repressor, this function is called as a negative regulation. So in negative regulation, a repressor protein, which is the function of a regulatory gene, it binds to the cis acting operator. Cis acting gene is the operator, and it turns off the transcription. So transcription is totally turned off. And now if there is a need for the removal of this repressor, there will be an activator, most of an inducer. Most of the time the inducer is the substrate itself. We will see this example in lac operon. But here just understand, a repressor binds to the operator, does not allow the RNA polymerase to move into the structural genes, and this whole process is always stopped. That is called as negative regulation. Now in positive regulation, what happens? At the site of the as you can see here, promoter, where the polymerase binds, at the site of the promoter the polymerase binds, there is a protein which is not a repressor, but it is an inducer, enhancer, let us say. It is a positive regulation. What it does, it attaches to the RNA polymerase and it acts as a accelerator. It enhances the factors, they interact with the RNA polymerase. As you can see here, if this is the promoter and structural gene starts here, the activators are also produced by regulatory genes. Regulatory genes can produce activators or repressors. So when activators are again protein molecules, they are formed, they go and attach to the RNA polymerase, they accelerate, means accelerate the reaction, enhance the reaction. These will be called as activators or enhancers. So in positive regulation, a trans-acting factor, what is a trans-acting factor? It is a protein itself, binds to the cis-acting gene. A trans-acting gene produces a trans-acting factor which binds to the cis-acting gene, that is the promoter region, polymerase, along with a enhancer or activator, and it initiates the transcription process. We will see this in lac operon and tryptophan. So this is positive regulation. Now we have two terminologies, negative regulation and positive regulation. But you see, this can also be called as an inducible system and a repressible system. This, what you have seen in positive regulation, is an inducible system, whereas what you have seen in the repressor back here in the negative regulation, where a repressor is acting, it is called as a repressible system. Now if you take into consideration, you see a negative control and a positive control, and inducible system versus a repressible system, you can have, try to understand, it is very easy. An inducible system and a repressible system combined with negative control and positive control can give rise to negative and inducible. The first case, we have four blocks here, four corners. See here, first is negative control. What is negative control? See the figure. I will zoom into the figures once again. Now this is the last slide for me to explain you about the type of inducible and repressible regulation of gene expression. Gene expression is protein synthesis. Regulation of gene expression is control of protein synthesis, that is the deciding the concentration of the protein. Regulation of gene expression can take place either of the four, one way, either of the four types. Which are the four types? Negative control, positive control, inducer and repressor. So if you just make a combination 2 by 2 table, negative control plus induction. So what is negative control? A repressor is formed. A repressor binds at the site of the operator, and at that operator point it does not allow the polymerase to move. That is negative. Now what is negative plus induction? In this there is another inducer, a protein molecule or the substrate molecule, it can be anything, which inactivates the repressor. It binds to the repressor, it inactivates the repressor, it does not bind to the operator, and there is synthesis of structural genes. This is induction. Now induction but positive control, repressor is formed. That inducer, instead of inactivating the repressor, it activates the RNA polymerase. There is an active activator. Instead of inactivating the repressor, it activates the activator, and this activator accelerates the RNA polymerase. So these are the beautiful complex mechanisms present in prokaryotes. So what is happening? Inducer and a negative control, inducer and a positive control. Similarly, now see a repressor. Repressor and a positive control, repressor and a negative control. Okay, how does that thing happen? In the negative control, first is negative, repressor is synthesized. This repressor may bind to the operator site. This inactive repressor is activated by another co-repressor protein. This is a separate type of system, may be present in different bacteria. I am not saying that all these are present in only one type of bacteria or a cell or a plant or animal. No, these are different types which may exist across the life systems. In general, taking a bird's eye view of the whole process, these 4 types of regulation processes exist in nature. So repression, a negative repression, negative repressible system. Repressor is formed which is inactive basically. Okay, in the inducer it was active, but here in the repression it is inactive, which is made active by a co-repressor, and then it goes and binds at the site of operator. This is called as a repressible but negative system. Now repressible but a positive system is, there is an activator which is active, which is inactivated by the presence of a co-repressor, so that it is inactivated, and if it is inactivated it will not bind to the RNA polymerase and it will not carry out the protein synthesis. So in general, these are the ways by which the inducible and the repressible systems are positively and negatively controlled in the bacteria and the prokaryotes. So to summarize this, an inducible regulation, in an inducible regulation, the gene is regulated by the presence of a substrate called as an inducer. This inducer is usually the substrate of the main reaction. In a repressible system, which can be positive or negative, the gene is regulated by the product of an enzyme pathway, that is a co-repressor. So there is an inducer in the induction system and there is a co-repressor in the repression system. Regulatory circuits or the regulatory mechanisms in all the living systems, they can be designed from all possible 4 combinations of this positive, negative, inducible and repressible system. So gene regulation in all the cells across the biological life can utilize any one of these mechanisms, and resulting into 4 types of gene regulation mechanisms, that is negative but inducible system, negative but repressible system, positive yet inducible system, and positive yet repressible system. So we will see all these types in our next lecture when we will be dealing with the lac operon and the tryptophan operon. But before that, we need to understand how the regulation takes place at the level of transcription and how the regulation is controlled at the level of translation also. So these are the 4 possible types. You can pause the slide here, go into details of this beautifully explained figure. Everything will be clear. Just remember that in inducer system there is a inducer, induction system there is a inducible system, there is a inducer. In a repressible system there is a repressor, co-repressor or a repressor, that is coming from 2 different areas but same, are here inducer or activator, that is coming from two different systems but one and the same. So inducible system involves inducer, repressible system involves a repressor. So that is all for with respect to the basic concepts of gene regulation. So having said that, the next lecture we will be studying gene regulation at the transcriptional level, how the enhancers, activators, etc., how the gene is regulated, means it is decided at the level of mRNA synthesis only whether it is needed to proceed further or not. If, for example, if there is lactose in the medium and that lactose needs to be utilized, you need enzymes to degrade lactose. For how long the enzymes will be needed? As long as lactose is present in the medium. A time will come when lactose will be over. After degradation over. Then this lactose enzyme machinery has to be stopped. How does it get stopped? That is gene regulation. We will see that. And that can be regulated at the level of transcription, means no need for the mRNA to be synthesized, or it can be regulated at the level of translation. During all these processes there are activators, enhancers, inducers, repressors, which are all synthesized and acted on the segment of the gene which is called as a operon. So this is the beauty of genetics. I want you to go through this lecture once again in case you have not got it for the first time. Listen to this lecture a couple of times and I am sure that you will understand the beauty of the regulation of gene expression, the beauty of protein synthesis taking place in the bacterial or the prokaryotic systems. So in my next lecture we will venture into controlling the protein synthesis at the level of mRNA synthesis, which is called as gene regulation at the transcription level. So till that time, I hope you understand and comprehend these beautiful molecular biology processes, and each lecture becomes a stepping stone for you to gradually understand the next point or the next level. That is what graduation is all about. That is how you become proper and appropriate, well qualified graduates for this science society. So all the best for that. Study well, understand well, and if you have any doubts and questions, ask me. At the end of this video, I have given some links which will help you to understand better and take out the notes. Take help of that and enjoy. Thank you. See you soon.